In this video, I'll discuss the levels of protein structure. And so there are different levels, and each one has its own purpose. Primary level simply tells what's present. That is also known as the sequence or the order of amino acids. Secondary level is when you see hydrogen bonds. These hydrogen bonds occur between the backbone of the protein. The protein backbone was obtained in the primary level, and that is simply when you see that pattern of the NCC from the amino acids. So the amine of one, the alpha carbon, and the carboxylate group of one, and then you'll see that same pattern repeating itself after the protein has come together. So basically you'll see NCC where you have this hydrogen. So if this were the very first one, this would just be a protonated amine. You have your alpha carbon, then you have the carbonyl for the carboxylate, and you have the next one. Now it only has one hydrogen. You have your alpha carbon, and then you have your carboxylate. And this process will continue until you get to the very end. At the very end, you simply have the carboxylate. So this NCC, that pattern till you get to the very end, that represents your protein backbone. In the secondary structure, hydrogen bonds can occur between the amine of one end of the backbone, or one portion of the backbone, and the carboxylate of another portion. This is where you would see your alpha helix or the beta pleated sheet, which is simply referred to as a beta sheet. The tertiary level is when the protein folds. So this is where it gets its shape. So that's where the shape of your protein is determined. This is when you start to see different interactions between the side chains. So not the protein backbone, but the actual side chains. So these are like intermolecular forces. So you learned about intermolecular forces in 1151, such as London dispersion forces, dipole-dipole, hydrogen bonding, so on and so forth. This is when you start seeing those interactions in the tertiary level between the side chains. So the secondary level was the protein backbone, but the tertiary level is between the actual side chains. So for instance, let's say that you were told that you were looking at the interaction that would occur between, let's say, alanine and I'll just say phenylalanine. So alanine, and I'm gonna draw a little squiggly line just to show the actual backbone, the side chain. And so for phenylalanine, you have a CH2, and then you have a benzene ring. So the type of interaction that would occur between those two side chains is what we call, or what we see in the tertiary level. In order to determine the types of interaction, you have to look at the types of side chains. Think about the intermolecular forces. Alanine has a nonpolar side chain. Phenylalanine has a nonpolar side chain. So the interaction that you would see would be called a nonpolar, or you may see it referred to as a hydrophobic interaction. Remember, hydrophobic simply means that it fears water, so it's not going to mix with water. So it's nonpolar. Some other examples, you may see polar, which is also known as hydrophilic interactions. This would include what we would typically refer to as dipole-dipole. You could even have hydrogen bonding included in that. Sometimes you'll see the hydrogen bonding listed separately, even though it is part of the hydrophilic interaction. This is different from the hydrogen bonding that took place in the secondary level, because now we're talking about the side chains. You can also have salt bridges. Salt bridges occur between an ion and a polar side chain. So this is what you previously learned in 1151 as an ion dipole attraction force. And then you could also have a disulfide bridge. And this occurs to form a covalent bond between two sulfur. This is the result of two cysteine residues coming together. A quaternary may or may not be present. Quaternary just means two or more peptide chains that must be included to make that protein function as it should. Not every protein requires multiple chains, but for those that do, they would also have a quaternary structure. That quaternary structure is basically the same as the tertiary. It's just telling you that now you're looking at what happens between multiple chains. That quaternary would also have the primary and secondary, because we would be able to indicate what's present, and we will be able to see hydrogen bonds between the backbone. So basically, if you have the quaternary, you're going to see multiple primary, secondary, and tertiary levels for that specific protein. So again, the most challenging part about the levels of protein structure would be the tertiary. And so in order to determine which type, you're simply looking at those side chains and thinking about the interactions that would occur. The best way to approach this, classify those side chains. Are they polar or are they nonpolar? You can even ask yourself, are they ionic? So do I see actual charges? Because if they are both polar, then you're going to see your polar interactions. If they're both nonpolar, you're going to see your nonpolar interactions. Hydrogen bonds, remember, for hydrogen bonding to take place, you need to see hydrogen that is either bonded to nitrogen, oxygen, fluorine, or you also need to see non-bonding electrons on nitrogen, oxygen, fluorine. If those things are not present, then hydrogen bonding cannot occur. And then for a salt bridge, you just need opposite ions. So you need a positive and negative. And then the disulfide bridge, you need two cysteine. And so this is how you would identify the types of interactions that would exist. But let's say, one more example, just to make sure you got it. So let's say that you were looking at the interaction that would occur between, I say, aspartic acid and, let's say, lysine. So aspartic acid, I'll just go ahead and draw out the actual amino acids just so that you can see those structures. I'll even expand the carboxylate group. So aspartic acid, you have this. And then we can look at structure for lysine as well. So remember, when you're looking at these amino acids, you're dealing with an alpha carbon, and then you have your amine, your carboxylic acid group, you have your hydrogen. The only thing that's different is simply this R group. This is how we classify the amino acids. This is how we determine the type of interaction that would occur. So we have aspartic acid, which is in the ionic form, has a negative charge. We have lysine, which is in the ionic form and has a positive charge. So the interaction that would occur between these would represent a salt bridge. And we know that because we have the positive and the negative. If they were both positive or both negative, we wouldn't have a salt bridge. If one was positive and the other one was neutrally charged, we would not have a salt bridge. It must have a positive and a negative in order for the salt bridge to be present.